Okay, so I will directly start. Multi-drug resistant bacteria are one of the major public health threats, and even though most of these bacteria reside mainly asymptomatic in the gut, they can easily bloom up to high densities after certain perturbation, for example, antibiotic intake. They then subsequently leading to an increased risk, especially in vulnerable patient populations. And also on the other hand, gut carriage of multi-drug resistant pathogens higher the risk for community spread of this multi-drug resistance. The currently limited treatment options and the increase of multi and pan resistance highlights the urgent need for alternative antibiotic-free treatment options. Normally, the healthy gut microbiome serves as a valid barrier against multi-drug resistant pathogens and harbors therefore a huge reservoir of potentially new probiotic species which can help to re-establish colonization resistance after medical perturbations. Specifically, we are aiming to address two questions. First, which bacteria are crucial to maintain and re-establish colonization resistance against MDR pathogens, and how do commensal bacteria maintain colonization resistance? Recent studies already highlighted multiple ways how commensal bacteria provide direct colonization resistance, for example, by competition for nutrients or other substrates like oxygen, or via the production of inhibitory compounds such as short-chain fatty acids. In order to characterize the natural levels of colonization resistance in the healthy population, we developed a simplified ex vivo screening system. For that, we spiked fresh fecal samples from a cohort of adult and children with 10 to the power of 6 CFUs of a multi-drug resistant Klebsiella pneumoniae isolate, and incubated then the samples for 24 hours under anaerobic condition and then recovered viable amounts of Klebsiella on selective agar plates. Interestingly, the samples from both cohorts showed high level of variability with up to 100,000-fold changes in the in situ growth of the pathogen. And in both cohorts, we saw the majority of samples had an intermediately high protective level, but we were particularly interested in those samples that could highly suppress the outgrowth of multi-drug resistant Klebsiella pneumonia. So we were asking ourselves what makes these protected samples special. We looked at multiple known influencing factors, for example, the overall community structure, variation in pH, and the levels of specific metabolites, for example, short-chain fatty acids, but could not find a clear correlation that fit to the outcome of the ex vivo assay in both cohorts, except the level of facultative anaerobes and specifically enterobacteria, which were correlated with protection in that way that the protected samples had higher levels of these specific bacteria. We then specifically looked at the two most protected samples and could isolate commensal Klebsiella oxytoca strains. We then further isolated these commensal Klebsiella oxytocas from different donors and found in total 23 isolates, which came, except for one isolates, only from protected and intermediate donors. The genomic analysis then revealed that these Klebsiella oxytoca strains form a unique cluster within the whole Klebsiella oxytoca species complex, discriminating them from nosocomial reference isolates and also from recently described commensal Klebsiella michiganensis strain, which has been shown to be protective against E. coli and also Salmonella. To investigate whether the protective properties of this human commensal Klebsiella oxytoca isolate are also visible in vivo, we destroyed the natural colonization resistance using antibiotics and pretreated SPF mice with this commensal Klebsiella oxytoca isolate before we subsequently colonized the mice with Klebsiella oxytoca. And you can see here on the colonization kinetic graph that those mice that harbored Klebsiella oxytoca in the microbiota, here indicated as blue dots, could clear out Klebsiella pneumonia in comparison to the control mice. And here in terms of levels. The clearance rates were up to 80% in those mice receiving Klebsiella oxytoca beforehand. Next, to assess whether Klebsiella oxytoca is also able to outcompete Klebsiella pneumonia in a co-colonization model or even in a therapeutic manner, we colonized SPF mice in parallel with Klebsiella pneumonia and Klebsiella oxytoca, or after 3 days of Klebsiella pneumonia colonization. And again, Klebsiella oxytoca pre-treated or treated mice could largely outcompete Klebsiella pneumonia from the gut. So this showed that there is a broad principle that Klebsiella oxytoca can mediate colonization resistance, and we were interested to see whether this is also true for complex human communities. And to test this, we took stool samples of 4 different susceptible donors, which were identified in this ex vivo screening assay, and transplanted the stool into germ-free mice. After 2 weeks of engraftment, 1 mice per donor was given, in addition, Klebsiella oxytoca, whereas the other mouse was left untreated before subsequently all of the mice were again colonized with Klebsiella pneumonia. And you can see here on the graph, which is quite colorful, that those mice that had Klebsiella oxytoca in the microbiota could greatly outcompete Klebsiella oxytoca, here indicated as dashed lines, in comparison to the respective control mice, here in the same colors for the 4 different donors, but with solid lines. In total, 3 out of 4 could outcompete Klebsiella pneumonia completely from the gut, and also the 4th donor mice could greatly reduce Klebsiella pneumonia CFUs. In the next step, we then wanted to see whether both species would maybe occupy the same niche in the gut and are thereby competing. For this, we performed a fish analysis of the cecum of Klebsiella oxytoca and Klebsiella pneumonia colonized mice, and saw that both Klebsiella species seem to be distributed in the lumen, suggesting that they may compete for similar nutrients. To test this hypothesis, we isolated cecal content and spiked Klebsiella pneumonia together with the different Klebsiella oxytoca isolates or an E. coli into this content, and incubated the sample for 24 hours under anaerobic conditions until we recovered the CFUs of Klebsiella pneumonia. And you can see here that almost all of the Klebsiella oxytoca isolates lead to a reduction from 100 to 1,000-fold in comparison to those samples that only harbored Klebsiella pneumonia, or those samples with an E. coli isolate or the MR01 isolate, which was coming from a susceptible donor. So this was hinting that both the Klebsiella pneumonia and Klebsiella oxytoca are indeed competing for the same substrate. And to prove this, we performed a comprehensive phenotypic microarray to screen for the utilization capacity of in total 190 carbon sources for selected protective strains, which are indicated here on the left in comparison to non-protective strains, and the Klebsiella pneumonia here on the right. We found that in total, Klebsiella pneumonia could use 56 different carbon sources, but the protective Klebsiella oxytoca could in total use double the amount and covered 55 of 56 of these carbon sources, almost covering the whole pattern. And this was also seen for the other protective Klebsiella oxytoca isolate. Interestingly, we found only 1 carbon source that could be exclusively utilized by Klebsiella pneumonia, this carbon source lactulose. And to prove if carbon source competition is really the causal mechanism, we tried to diminish the protective effect of Klebsiella oxytoca by supplementing mice lactulose. So we performed the same experiment as I described in the beginning, but fed the mice via the drinking water 10% lactulose during the Klebsiella oxytoca colonization until 2 weeks after Klebsiella pneumonia. And you can see here that those mice that got lactulose in addition and Klebsiella oxytoca could not clear out Klebsiella pneumonia until the end of the sugar supplementation. Only after the ending of the sugar supplementation, also those mice having Klebsiella oxytoca could then clear out Klebsiella pneumonia, indicating that that carbon source competition is really an important mechanism. To summarize the main finding of these studies, we found that ex vivo colonization against Klebsiella pneumonia is highly variable between healthy individuals, and we isolated diverse commensal Klebsiella oxytoca strains from particularly protected individuals that could establish colonization resistance against Klebsiella pneumonia in variable microbiota situations. Further experiments then revealed that specifically beta-glucosidic sugars play an important role in this interspecies competition, which is mediated at least partially via a specific gene, the Cas8 gene that we found in protective Klebsiella oxytoca isolate. Another point that I wasn't mentioned until here is that we found that this colonization resistance is dependent on 3 additional bacteria from the Firmicutes phylum that can further restrict nutrient availability in the gut. Next, we are aiming to address 2 questions. First, which other genes are important for sugar restriction besides this newly identified Cas8 gene, and can Cas8 serve as a biomarker for other protective bacteria? With that, I'm at the end of my talk. I would like to thank for your attention and would like to thank all the people that were involved in the study.